Welcome to the video for day 3 of the ChIP experiment. Your samples will be at 65 degrees Celsius. Spin down your samples to pellet any insoluble material. Your tubes, especially the input tube, may have a white pellet at the bottom of the tube. Use a pipette to remove the supernatant to a fresh, labeled microcentrifuge tube for each of the 3 samples. Add the DNA bind buffer, pipette to mix, and then load the sample onto the appropriate labeled spin column. Repeat for the other 2 samples. Use the microcentrifuge to spin the samples through the spin columns. After centrifugation, discard the flow-through. Apply the wash buffer to each column and repeat the centrifugation. Discard the flow-through. Repeat the wash step 1 time. As you discard the flow-through from the second wash, move the spin column to a fresh, labeled microcentrifuge tube. Obtain a tube of pre-warmed elution buffer. Pipette the buffer onto the center of the spin column. Incubate the buffer on the column for 5 minutes. Centrifuge the tubes to elute the DNA off of the column. After centrifugation, remove and discard the spin column. The DNA will remain in the buffer in the tubes. Set up an ice bucket with all of the reagents and tubes that you will need to set up your qPCR reactions. Create a master mix by combining the SSO Advanced qPCR mix with the purified water. Distribute the master mix into each of the 24 reaction tubes. Next, add each primer to the appropriate tubes. add water to the 3 no primer control tubes. Next, add your purified DNA samples to the appropriate tubes. Now, transfer your reactions into the appropriate 96 well plate. Be sure to note which samples you are placing into each well. Take your plate into the instrument room and seal the top of the plate. Make sure you press the seal down around the edges and across the tops of the wells to prevent evaporation. On the computer, select the CFX Manager software. Choose a user-defined run, and select the existing ChIP qPCR protocol. Check the plate layout to ensure that the appropriate wells are selected and that cybergreen data is being collected. On the next screen, click to Open Lid. Once the lid has fully opened, place your 96 well plate into the instrument. You can click the button on the front of the instrument or in the software to close the lid. Once the lid is closed, you can select Start Run. The program will ask you to select where you want your files saved. The program will then switch to initializing. Check that the light on the instrument is on, and the program is proceeding with the run. Upon completion of the run, a window will display the amplification curves and the flat line for the calculated threshold. You can highlight specific wells to only display certain curves, or hover over each well or curve to highlight the corresponding curve or well. For example, selecting one row will display all of the curves from one DNA template. The program will also give numerical results for the CQ, or cycle at which the amplification crossed the threshold, for each well. It will also display the melt curve analysis. This is a control to ensure that only one product was produced with each primer set, as indicated by the presence of a single peak. These results can be exported into an Excel file format. Thanks for watching!